Hello, thanks for coming to this week's video lecture. Today we're going to be talking about chapter 4, where we go over prokaryotic cellular structures and their functions. And we'll talk about chapter 7, where we go over the metabolism, uh, metabolic pathways of, sorry, we'll go over briefly metabolic pathways of like nutritional requirements, how they can affect how bacteria grow in different conditions, and then how the environment itself can change how a bacteria can grow. With that, we can get started. Something to remember is that prokaryotes don't have membrane-bound organelles. And so something that's the most striking is that they lack a nucleus. And instead, they have what's known as a nucleoid region, where you have their chromosome, which is a circular piece of DNA that has been condensed down into a tight bundle into a region of the cell. And instead of having a barrier like a nucleus and having an envelope that surrounds it, instead, we're relying on kind of separation just by compacting and a compartmentalization rather than solid barriers. Inside the cell, you have the cytosol, which is a thick, very dense liquid that is made up of water, and then it's filled with thousands of proteins, thousands of sugars and chemicals and compounds, and I'll say enzymes too, but those are also proteins, so they're kind of the same thing. But this is like thousands and thousands of molecules just butted up against each other, where there's thousands of collisions happening just every second. This is how the chemistry is happening so fast, and all of these molecules are moving very randomly, and this is what's known as like Brownian motion. And so if you like watched a specific molecule, like if you was like, this one, I want to watch it, you could see it and it moves around, and it'll move around kind of just randomly. And that's kind of that's what Brownian motion is. And that's what's happening within these cells. And that's how these molecules at this small scale kind of operate, and how if you have two molecules and they're supposed to be like interacting in some kind of chemical reaction, they'll be moving around. And if they hit in the right way, at the right orientation, at the same time, they can overcome some kind of energy barrier to trigger that reaction to occur. That's how this chemistry is happening. It happens so fast. It's just mind-boggling. Anyway, within the cytosol, you have all of those proteins that are facilitating the biochemistry within the cell. And then you have the ribosomes. And these ribosomes take the RNA that is transcribed off of the DNA and translate proteins. And so these ribosomes are what make all of the proteins that are within the cytosol. And also, there are ribosomes on the very edge here by the cytoplasmic membrane, or the inner membrane, that are specifically there and localized there to kind of help make proteins that are going to be secreted outside of the cell. Excuse me. So then the other things that you have within the cell are inclusion bodies, and these are just kind of aggregates where things have just gotten, there's a lot of something that you don't really need, and you don't really need it in everywhere. You push it and compartmentalize it into something very specific. So if you had some kind of waste, if you could put it into one little spot, that would be great. And so that is kind of the idea, is that these are aggregates of molecules, whether they be stores for nutrients, whether they be waste, whether they be toxins, whatever that is, or fat droplets. You also have plasmids, which are extra chromosomal DNA. And these plasmids are typically not only found in the nucleoid region, but also in the cytoplasm itself. But these are kind of little cassettes of genes that boost survivability, or like give you some kind of like advantage to living life. This can be, I can now produce a new toxin. I can now digest some kind of new nutrient. I can survive some kind of new damage. And so these plasmids are used for that, and they can actually be transmitted from bacteria to bacteria. And so if you have something that's like antibiotic resistance, it can transmit from one bacterial species into a new bacterial species through these plasmids because they're extra chromosomal and they have an easier time being transferred through established mechanisms that we'll talk about. So I kind of mentioned that the cytosol is wrapped up with the inner membrane, or the the cytoplasmic membrane. And this is the membrane that kind of holds it all there. It has a connection with the cytoskeleton. The cytoskeleton is made up of actin filaments, and these provide a little bit of support. They kind of give you something to hold on to. This helps with like transporting certain proteins to certain places. It also helps to kind of give a little bit of support for any pressure on the outside. But more so, the cell wall adds support and structure to the bacteria. So the cell wall is made up of peptidoglycan, and we'll talk about that. It's a sugar protein. It's a protein that's conjugated with sugars, and it creates a really strong network lattice that surrounds the cytoplasmic membrane, and it provides a lot more support and protects from like osmotic pressure and things like that. We then have, in some species, an outer membrane, and this outer membrane is an extra layer of protection from the environment. And then you also have some other other structures that aren't seen in all bacteria, like the S layer, which is a sugar protein layer that is helped for which is like triggered in response to like threat, and it helps to like defend yourself from foreign foreign adversaries. It helps protect yourself from like the immune system if you are in like the body, for instance. So then you have the fimbriae, and the fimbriae are these hair-like structures that protrude out of the bacteria, and they are kind of help with attachment. And so you can kind of think of them kind of like a Velcro, where they kind of settle in and are able to attach to whatever that surface is. And they're able to probably produce like intramolecular forces or things that kind of actually hold on there. And those help with attaching. And so when you're in the middle of trying to like grow and establish, you want to find a spot and you want to attach and you want to hold yourself there. And we'll learn about that attachment. That's a stage of establishing infection. And so these fimbriae help to do that. And there are other things like the slime layer or the capsule, which is kind of a looser sugar protein matrix. And it helps with like mobility to help move the protein. Help move the bacteria, kind of like a slug, essentially, where the slime layer kind of helps allow the bacteria to glide on a surface. But it also can help attach to that surface. And then it also helps from protection from the environment. And so, excuse me. And so, where was I? We were talking about the fimbriae, how they're good with attachment. The slime layer also good for attachment. The capsule's good for attachment. And then we have the pilus or the pili, depending if it's singular or plural, this is also called the F pilus or the fertile pilus. It kind of depends on who you're talking to. But essentially, this is a hollow tube that can connect two bacterias together. And this allows us to transmit bacteria between one another. And so this is where those plasmids are able to be, this is what's called conjugation. So your two bacteria can conjugate together, and it allows for the genetic transfer of transfer of genetic material. The easiest thing is plasmids because they're small, they're packageable, and they can move through it very quickly. Now, if this conjugation lasts for a really long time, what's really cool is there are instances where the genome itself from the nucleoid region will actually start to be transcribed or replicated and then sent through this conjugation bridge. And so you then have like it's trying to duplicate its entire chromosome into this other bacteria, which is crazy. And usually this is not successful, but what you'll see is there'll be times where you'll have a good sized chunk of a different species of bacteria in a new species. And it's because there are these conjugation events that have happened. And then that new introduced DNA gets integrated into the old DNA. And so this is a driver of genetic change over time, though very spontaneous. You also have these plasmids, which are like a lot more like a lot more common, happens all the time. This is a lot faster and a lot more common. So plasmids are problematic because like you'll hear about like the spread of antibiotic resistant bacteria. It is because this conjugation process occurs. And so like if you had a bacteria that had some kind of antibiotic resistance and you killed it, it's just shredded up bacteria. That plasmid's still floating around in that shredded carcass. And so if a new bacteria comes up, there are ways of transfection, of pulling that plasmid into the bacteria, and that new bacteria pulling that plasmid from the dead carcass and being able to get that like antibiotic resistance. And so that's why it's really hard to fight, and like some like scary to fight these like to fight these antibiotic resistant strains of these antibiotic resistant strains of bacteria, is because they do pose an actual like risk, and it is like a little wild to think about. Lastly, the last structure to think about the last structure we're going to talk about is, I guess it's not, but is the flagellum. The flagellum is like a whip-like appendage that helps with motility. And then you have endospore, which is kind of like an inclusion or a granule that forms inside of the cell. And we'll talk about how it is kind of like a doomsday bunker for once the cell dies, this endospore can sustain itself in incredibly harsh conditions and regerminate over many years later and make a new bacteria from that endospore. It's kind of like a vault to keep itself safe. So now that I went into all those details over just like the first slide, let's get started. So, so we have those structures that are on the outside of the cell. We went from the inside outside. Now we're going to go from the outside inward. And those appendages, those structures that are on the outside of the cell are the flagella. That's that whip-like appendage that helps with motility. It swirls around. You have the fimbriae, which are hair-like protrusions, which help with attachment. And then you have pili, which help with conjugation and the transfer of genetic material. When we talk about the actual surface of the bacteria, you have the surface coating, the capsule, the glycocalyx, the slime layer. In different species, it's called different things because they're made up of different things. But overall, they're protecting from evaporation. They're protecting from UV. They're protecting from host immune systems. They're providing protection from maybe other chemical agents from other bacteria. And then you have inside of that, you have the cell envelope, which is made up of either that outer membrane, if present, a cell wall, and a cell wall, and then an inner cell membrane or the cytoplasmic membrane or the inner membrane. And so the fimbriae is and pili are good for attachment and transfer, while the flagella, slime layer, the flagella and slime layer are good for movement and motility. Again, that slime layer is like a really like, it's kind of like mucusy, and so it can kind of slide through, slide with it. So the flagella is this whip-like appendage that rotates like a propeller, and it is made up of a filament. These proteins are repeating units, and so they are shaped like this, and then they stack like that into a very long strand, and then they bundle those strands to make a filament. And this filament is then is protruding into the environment. It then has a hook and a basal body. This basal body is actually what anchors it into the the bacteria's membrane. And so the basal body, this is where the actual like motor happens, and actual turning mechanism is occurring. And then you have, and this turns the filament, and it causes this whip-like motion. And so you can have these flagella located on a couple of different places. You can either have them all over the surface, or you can have them on specific places. If they're only on the ends or on a end, they're called polar. And if they are in bunches, they're called lymphotrachous. If there's one flagella, it's monotrachous. And if there's and then there's also like ditrachous and things like that. And then if you have flagella all over the place, you have peritrachous cells. And so if you look here, we have monotrachous, where we have one flagella that's on the one end. You have lophotrachous, where you have a bunch of flagella on one end. You have peritrachous, where they're all over the place. You have amphitrachous, where there's two bunches on either end. And so if you think about which one of these are polar, you can think about which ones are on the end. And so in this case, this one are on the end, this one's on the end, and these ones are on the end, while the peritrachous is not on the end. And so the answer would be these three here. And these flagella help to move the bacteria in what is known through chemotaxis, where we're following some kind of chemical signal either towards a stimulant or away from a stimulant. And so positive chemotaxis is if you're trying to follow towards the chemical signal. So this would be like if you're going after food. While negative chemotaxis would be trying to move away from the food. Sorry. I was eating and got distracted. Would be like moving away from the stimulus. And this would be kind of like if there's a toxin or some kind of stimulus that you don't like, you can move in the opposite direction that it occurs. And this is called negative chemotaxis. And chemotaxis, when you have a flagella, happens in what are known as runs and tumbles, where the flagella will rotate in one direction, and this will let you shoot forward in one direction. And then the flagella will rotate in the opposite direction. And what this causes you to do is kind of halt and kind of tumble And that random tumbling will reorient you and face you in a different direction, where you can then reorient the flagella and start running again. And this causes you to kind of go in kind of random motions. But when there's a stimulant, these tumbles are much more controlled, and it can allow you to actually move in the direction of your tumbling. It kind of makes a little bit more sense when you look at the peritrachus bacteria here, which is also what E. coli has, these kind of flagella like this. And so you can kind of see how if you twist in one direction, they all kind of work together, and then they have to all untwist, and it kind of, you could see how that kind of makes some maybe turbulence and maybe makes some tumbling if you kind of think about how stuff works in water when you, like, if you had like a, I don't know what's a strap and in water and you tried to like spin it or whatever. I feel like you would feel those forces. Oh my. These flagella can actually also be inside the bacteria. And so instead of being on the outside, we can actually put it within the space between the cell wall and the inner membrane, called the periplasmic space. This is the space between the cell wall and the inner membrane. And the flagella wrap around the bacteria in a spiral shape, and it causes, um, sorry, the flagella are, sorry, my brain's broken. Sorry, I have functional movement disorder, which causes my brain to forget how to function sometimes. But these flagella are in this space, and it causes the bacteria to have this corkscrew-like appearance and movement through the media. I'm sorry. One thing that you would, Helicobacter pylori is a spirochete bacteria. We then have fimbriae, and fimbriae is are those bristle-like appendages or hair-like appendages which help with attachment. This is a transmission electron microscopy image. And you can see kind of those fimbriae here and kind of how they're working to attach themselves to the intestinal lining. So sorry. I have bronchitis. So we have the pili, and the pili is helps with that, facilitates the transfer of genetic material. And so you can, and the process of that transfer is called conjugation. Yeah, it also, like, cytoplasmic material and like proteins and like nutrients and things would also like flow through those tubes as well. So there would be nutrient exchange and like just other things that would also be exchanged. But the biggest thing is that genetic material. You also have the fimbriae just visible in here. You can kind of see how that fimbriae is much more distinct than is distinct from the pili. And then you have the surface coating glycocalyc slime layer. And these are going to help you keep moisture within yourself, give you protection from the environment and the host immune system, and also in some instances help with mobility. And you can kind of see how this would kind of, here is the bacteria here, where you can have like a bacteria there and a bacteria there and a bacteria there. And once you have a biofilm, which is made up of similar things to this glycocalyx, you start to lose the definition of where is one bacteria start and where does one end. And this is the benefit of these kind of glycocalyxes and slime coats is that it helps to provide protection and from the immune system, because the immune system isn't going to be able to pick you out as much. That's kind of a naive way of thinking of the immune system, but it's fine for now. And so, but these glycocalyxes and things all work together to make biofilms. And these biofilms are things like plaques in your teeth. It can be plaques on catheters and like medical devices. And it can be biofilms on industrial, like on like agricultural equipment and like water treatment plants and like pumps, and like literally plaques form on everything in everywhere. I'm sure there would also be biofilms in your gut. I just haven't heard about them particularly, but I'm sure I'm sure that they exist. Excuse me. And so yeah, and so biofilms are they're also hard to get rid of. And so usually they are really hard to dissolve or dismantle, even with like chemical treatments. And so once they're there, it's hard to get rid of. And so with medical devices, this is like a big problem. For like water treatment plants, that would also be like a big problem, because you could have like catastrophic failures within your equipment. Like you would lose access of like being able to supply your like city with water or whatever. So then you have the cell envelope, which has that outer membrane. It has the cell wall, and then it has the inner cell wall. So it has the outer cell wall, sorry, the outer membrane, the cell wall, and the inner membrane. And sometimes that inner membrane isn't considered to be a part of the cell envelope. But there are two types of these cell envelope structures. There's a gram-negative and a gram-positive. And Hans Christian Gram developed this technique of differentiating the structure of these nuclear envelopes by a differential dye technique, where you do a series of dye and decolorization steps to see how thick, essentially, and how soluble the membrane is to certain dyes. And one type ends up one color and one type ends up another color. So you can easily look at it and say, well, easy, it's purple and pink is the two different colors. And so like there's quite a little bit of overlap between purple and pink. And so it's sometimes, well, purple and pink, pink and blue, depends on how much color you use. But either way, purple and pink. But it's relatively easy to distinguish. It's an easy technique to do, and it's an easy thing to distinguish. And we're going to do it a lot in lab. So we'll go over that in lab, like the procedure of it. But the biggest thing is it's looking at the structure of these cell walls. And we already talked about how these cell walls are made up of the peptidoglycan protein, sugar. And so this is a protein that has been conjugated with a sugar. Conjugation just is a fancy name as they're bonded together relatively tightly. So you're not going to just like rip them apart easily. Peptidoglycan is the main component of the cell wall, and it provides that structure to the bacteria. We'll get back to the gram stuff later, about like what the colors are and things and how you actually do the procedure in lab. But yeah, it's important to know the structure that we're working with when you're handling like a specific bacteria, because the treatment for that bacteria heavily depends on that structure because that cell wall being incredibly thick or much thinner is like the thickness of that cell wall and the components of the cell envelope kind of make it where some drugs are more effective than others, depending on that structure. I don't understand why they waited until the very last, like the very end, to show you these structures, but sorry, I didn't think about reverse in the order for yeah. But this is the structure of a gram positive and a gram negative, where the gram negative have three layers, where you have that outer membrane, that cell wall, and that inner membrane. And that cell wall is really thin in negative. The thin cell wall for the negative, or a narrow cell wall for negative. With positive, positive stains purple, just so you know. And it has a very thick peptidoglycan cell wall, and it only has an inner membrane and it lacks that outer membrane. So it has two layers. It has that inner membrane again, and a thick peptidoglycan cell wall. Something to note is that periplasmic space that we saw with that spirochete bacteria with the flagella in between the membrane and the cell wall. It was in that periplasmic space. And that periplasmic space is between that yellow and brown line, that inner membrane and that thin cell wall for the negative. The negatives have a larger periplasmic space typically than the positives do. It's not super important that you know that, but it is important. It is like, it's a fact to know if you want. What's important about this space is it's one of the few locations that is a membrane bound, like it's essentially a membrane bound compartment that the bacteria is going to utilize for its metabolism of creating energy and doing the electron transport chain and respiration. And we'll talk about those later. But just know that, like, sorry, I forgot where I was going. But just know that this space is being utilized very specifically, like is being leveraged to do biochemical like functions and metabolic functions. So with the gram positive, we have that thick cell wall, and that thick cell wall usually has different acidic sugar molecules and as well as proteins embedded throughout the membranes that help facilitate signaling, help facilitate transport of different chemicals in and out, or with, for instance, the metabolic pathway that we'll talk about later. The gram negative, on the other hand, it has the three layers, right? It has that inner membrane, the thin cell wall or that narrow cell wall, and that outer membrane. And that outer membrane is special because it has porin proteins, which help easy inflow and outflow of small particles. It also has lipopolysaccharides, or what is usually shortened to LPS. And these LPSs are lipids that are conjugated with sugars, and they're polysaccharides. So it's branched sugars. So it's like sugars that have been bound together. And then they're stuck onto a lipid. And these lipid polysaccharides are typically fairly immunogenic, so they cause an immune reaction, and they're typically considered to be toxins. So a lot of the time, they can cause symptoms of when they're released into the body. And in research, they're used a lot to trigger an immune response. an innate immune response. So you have the gram-positive, which have the thick cell wall and the inner membrane. Then you have the gram-negatives, which have the outer membrane, the thin cell wall, and then the inner membrane. And something that's to keep in mind, we talked about how the gram-negatives have LPS, while the gram-positives have these slightly acidic sugar molecules. So you can kind of see how the structure is different between the two, and you can see the periplasmic space here on both of them. So not all bacteria have these cell walls made of peptidoglycan. Mycobacterium usually have mycolic acid that create like a waxy coating around it. You also have mycoplasmas, which have no cell wall, so they have a lot more abnormal shapes to them. So Mycoplasma pneumoniae, which causes walking pneumonia, is one that kind of has obscure shapes because it doesn't have the structure of the cell wall to keep it rigid into a uniform shape. So then you have that inner membrane or the cell membrane. And this cell membrane, like we saw in those previous slides, have these associated proteins embedded in and throughout the membrane. And this is to help us communicate from the inside and the outside of our cell. So you can think of having this, for instance, this integrated protein here. Let's say you could have something that binds here, and that could cause a change on the inside, and that change on the inside could then get detected. And that's how a signal transduction occurs across a plasma membrane. You also have things that are like binding sites. So for instance, something binds here and that would release this protein to go do something, for instance. You have transport proteins, which allow inflow and or like influx or efflux of certain compounds, certain molecules or certain atoms or ions. It depends on the pores, or it depends on the proteins to determine like what can and what can't come and go through these membranes. Typically, charged molecules can't just flow through the membrane, but neutral molecules, nonpolar molecules can flow in and out just fine. Sorry. I think I need to go a little bit faster. So the genetic material, you have the chromosomes, which is in the nucleoid region, which are tightly compacted in that nucleoid region. You have plasmids, which are free floating in the cytosol, which carry those like extra bits of survivability boosts, those little like cheat codes that you could use. You have ribosomes, and ribosomes are to make the proteins that you need. While the genetic material holds the information to make the proteins. So DNA here. I don't know if you guys know this, but DNA. It's called the central dogma. This is unit 2. DNA goes to RNA, it goes to protein. It's very important in molecular biology. It will also be very important in unit 2. Protein. Yeah, whatever. I don't even know if I spelled that right on that time, but it's fine. So the genetic material, these are DNA, and they hold the information. They hold our genes, and they get transcribed into RNA, and that RNA then holds just like 1 or 2 genes, and then those RNAs get turned into proteins by the ribosome. Okay, so the ribosome is making that protein. And then you have the storage bodies like inclusion and granules, which are just like aggregates of different like nutrients or compounds that you're not really needing to be all around. You have the cytoskeleton, which is providing support and helping with like internal transit of molecules. You have the endospore, which provides an extra layer of protection in case of existential threat and horrid environmental conditions. So we'll talk about we talked about the chromosome, it being in the nucleoid region, and that's kind of in a like compartment, but it's not membrane bound. They're like it's just kind of like a tangled mess just in the middle. And then if you need a gene for something, it'll kind of sometimes it'll kind of come out and it'll loop out. And then this is where outside, right on the edge, is where that transcription and things happen. And then that's kind of how it's managed. So then you have plasmids. We talked about that. We have the ribosome. Those ribosomes make the proteins using the RNA that is pulled from that is transcribed off of the DNA after it kind of loops out like that from the nucleoid region. And then the RNA is fed through the ribosome, and we'll talk about that again in a different unit. And it makes a protein through a couple of steps, and that protein folds up and can go do some work. Now, ribosomes are made up of two subunits. You have a top subunit and a bottom subunit. And for bacteria, it is a 50S large subunit and a 30S small subunit. Now, and then when you put them together, it equals a 70S subunit. And so these units, it would be really cool if they added up, but they don't, unfortunately. So what this is these are units of sedimentation. And so this uses a technique called ultracentrifugation, or analytical ultracentrifugation, or ultracentrifugation. But what you do is you you take a tube and you fill it up with different concentrations of sugar, where you have incredibly high concentrations of sugar to lower and lower concentrations of sugar. And when you spin your sample down, depending on the size and the density, you can kind of think of it like one of those like temperature thermometers, where it like, depending on the temperature, certain certain degrees will be like in different places. Here. Buoyancy thermometer. You can think of these. It's kind of like that. But it is so you have this gradient of sample, and you put a force, you spin it through. Sorry, these aren't really good demonstrations. I was really hoping. Give me a second. Okay, I found it's a sucrose gradient ultracentrifugation. And so you have these densities of sucrose, and when you put your sample on it and you spin it down, your samples will separate based off of those densities and sizes, and it corresponds with that concentration of sugar. And you can kind of see how that looks in real life here, comparatively to like the cartoon drawing. So they did this with the ribosome, and the small subunit settles at that 30S mark, the large at 50, and then when you do the whole, it's at 70. Now, what's frustrating is, it's not really frustrating, but eukaryotic cells have different ribosomal subunits. And so these 70S, 50S, 30S markings are going to be different nomenclature when you're talking about eukaryotic cells and prokaryotic cells. So it's really important to know that. Now, another thing that's really cool is these ribosomes are actually made up of proteins and RNA that are all folded up together. And so the ribosome structure is incredibly detailed and complex. This is what the ribosome looks like. You have that large subunit and that small subunit. And you can see the blobby, well, it's almost impossible to see what's what. So the green turny bits, that's protein. The little blobby bits that are tan and gray, those are RNA molecules. And those are working together in this like machine that reads in RNA and spits out protein. And I just want to say for future, this RNA is different than the RNA that it's reading. So this is rRNA, while what it's reading is mRNA. But that's next lecture, next unit, I mean. So then you have those inclusion bodies. We talked about those. We have the cytoskeleton that provides stability. Now the endospores. So the endospores are in a response to unfavorable conditions. You don't have enough nutrients. You have some kind of toxin. You have DNA damage. There's no water, and you dehydrate. So endospores are primarily seen in Bacillus and Clostridium species, nearly exclusively, and usually you have an endospore stain that you can see this. So this is probably Clostridium techni because it has a club shape. And then it has an endospore that's stained green. And so these spores are incredibly impervious to being like killed or like inactivated. They can withstand heat and chemical treatments, and they can also withstand time. And so for a very long period of time, these endospores can still regerminate and regrow a bacterium from it. And so that bacteria will duplicate its DNA and then section it off and start to form up the layers of this endospore. And then it'll start to desiccate it until it's like fully mature. And then once the environment gets so severe that it actually kills the bacteria, the endospore can just sit around until it's ready. Sorry, I thought I wasn't recording. I was like, oh, dang it. Until it's ready to, until the environment is ready to host the bacteria again, and it can start to, it can start expressing the proteins it needs and starts to build the bacteria back up. It kind of is like, ta-da, it's done. I'm sure the process of from here to here is incredibly intriguing. However, the book doesn't go into it too much. So but this kind of gives you like a second life in case things go wrong. And so typically, like we said, Bacillus and Clostridium are the ones that are typically going to be producing these endospores. An example of this is Bacillus anthracis, which creates an endospore that's we use and we've named as, I guess we don't use it, but people have used it and we have named it anthrax. Bacillus anthracis is a bacteria that's found in the soil. Produces these spores that you can inhale, you can get them on your skin, and they can cause rashes and irritation and illness. You then have Clostridium techni, which causes tetanus. You have gangrene and botulism, Clostridium botulinum, which is found in food products quite a bit, and it produces its endospores because it's in food products. And endospores are so like hardy. It's a true risk when it comes to food safety. Then it's why, in part, we have to, for certain foods, add this additional pressurized steam at incredibly high temps for long periods of time to actually sterilize these foods for proper canning safety. With like high acid foods and things like that, you don't have to always steam them because those acids and things can protect you from these endospores, but I mean, can protect you from certain things, but when the endospores are potentially present, you have to kind of go up to a higher level of sterilization. So now we can talk about the shape of bacteria. So bacteria can have a few different shapes where you can have round, rod, or comma shaped, where the round are called coccus or cocci, where the rod are called bacillus or bacilli, and the comma shape are called vibrio or Vibrio or Clostridium or Vibrio as well. And so some people call them Clostridium, but they're Vibrio. Anyway, some more specialized ones are Spirillum and Spirochete, and that's the rigid spirals and the flexible spirals or helixes. The distinguishment isn't super important in our case. And then I haven't seen this before, but the branching filaments, where it's just kind of abnormal growths. You see this kind of branching structure in the mycelium of fungus, but you don't really, I haven't seen this in bacteria personally. And then we talked about how some bacteria don't have a cell membrane, and that causes them to, like Mycoplasma pneumoniae, don't have a cell membrane, a cell wall, and it doesn't create like a uniform shape for them. And so they're all different kinds of shapes. And this is what's called a pleomorphism. You can kind of see how they're all different shapes of all different kinds. So when we describe bacteria, we want to describe them by their shape and arrangement. The shape is how does one bacteria look, and the arrangement is how do they look together. And so bacteria go from one bacteria to two bacteria to multiple bacteria through what is known as binary fission or replication. And depending on how they replicate, it determines their overall shape, where they can either be chains, cubes, or clusters, where if they have only one plane of division, they can be diplococcus, where they split into chains of two, or they can continue to divide through that one plane of division and create these long chains of streptococcus, because it's a long chain of spheres. If you have multiple planes of division, you can get these tetrads, which are these packets of 4 or packets of 8 with sericina. And then if you have too many planes of division, you can get abnormal divisions that create these cluster-like looks. And this is what's called staphylococcus. And so a lot of the time you're going to be comparing staphylococcus to streptococcus. And so you need to understand how what these prefixes mean on how they look for the bacteria. And so for those were all for coccus bacteria, but for bacillus, you don't have those staph kind of, you don't have that staph, those clusters. Instead, you only have strepto and diplo and singles, where you have individual cells, diploid, where chains of 2, or strepto, or long chains. There are some bacteria that you're not going to see very much that have little special tendencies that cause their arrangements to be a little bit specialized. So here's what's known as a palisade arrangement, where there's a bit of a hinge region right next to the plane of division, and it creates these side-by-side what they call palisades, because it's like a palace armed guard or whatever, a palaceman lined up against each other like you would see in like some kind of royal hall or whatever. So then we utilize these shapes and the arrangements to identify the bacteria that we're working with. And so over the decades, microbiologists have come together to try to figure out how the best way to classify these bacteria are and how to identify them. And one of the biggest winners of the space is called Berge's Manual of Systematic Bacteriology. This is something that every single microbiology student has at least done some kind of identification using this manual, and we're going to also be doing that. And it used to be that every single microbiology lab would have one of these, like a copy of one of these. However, now it's digital, so you don't usually have solid copies of it, but it's a big book that just can separate your these bacteria into these different characteristics, and they separate it into different phyla and genus, just like you would with the classical classification system. And you use things like the its metabolic profile, like what chemicals can it use to, like what nutrients does it need to eat, and things like what is its cell wall structure, to identify these bacteria. So here is an example, or the list of the domains where, for instance, I don't know, let's see, there's probably one in here that we can talk about. If you're interested in looking at them, I would read them, but I'm not going to go over each individual one here. Cyanobacteria, these bacteria are incredibly famous for their photosynthetic properties and the impact that they had in early Earth, in like converting the atmosphere and like making it from like major CO2 to, or like into like sorry, their major contributions in like increasing the concentrations of oxygen in the environment is contributed to the cyanobacterias, which have many different kinds of bacteria and different kinds of photosynthesis that probably occurs there. They have different colors. And then you have like different bacteria that are found in thermal vents and things like that. Then you have the phyla, and these phyla talk about different specific ones, like here for bacteroides, like Clostridium, like Clostridium difficile, Clostridioides difficile. Sorry, the name has changed a couple of times on that one. It's a gram-negative anaerobic rod, and so it's found in the gut, and it causes gastrointestinal like problems. And so these these bacteroides have a gram-negative, so they have that inner cell wall. They have those three layers. They have the inner cell wall, the inner cell membrane, the cell wall, and it's thin. It's a narrow cell wall, and then an outer cell membrane. Let me try to do that a little bit smoother, right? So we have the bacteroides are gram-negative, which have those three layers, which are the inner membrane, the narrow cell wall, and the outer membrane. It also it's anaerobic, and we'll talk about how that means it likes to be without oxygen, and then it's rod shaped, so it's bacillus in shape. And you can see that bacillus shape in this figure here, where it has those rods. And it's also pink, and gram-negative is pink, but you haven't learned that yet. Finally, we can get over to chapter 7, where we can talk about nutrition and ecology and what affects microbial growth. So microbes need carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur in order to grow and make new proteins and biomolecules to sustain its life. So these are the principal building blocks of life, and so they're very essential and are typically considered essential nutrients. And you can consider macronutrients and micronutrients depending on the quantity that they are needed. So things like carbon, hydrogen, and oxygen are macronutrients because you need them for literally every single molecule. While, for instance, sulfur is a micronutrient because you only need it for some proteins. Then we have organic and inorganic, where organic nutrients have that carbon hydrogen have a carbon hydrogen framework. And this is why those carbon hydrogens are macronutrients. Um, an example of this is like methane. Another example would be like glucose. Inorganic would be like not having that carbon hydrogen. So water would be non-organic. Uh, one that's a little bit better would be like CO2. Oh, water is an example, so I'm not pushing myself a little too far. So, excuse me. Sorry about that. So what you have to think about is uh, different bacteria have different ways of getting these essential nutrients and bringing these macronutrients into themselves. And so do they get these nutrients from other organisms, or do they get them from inorganic sources like carbon dioxide? So for carbon, how do we build up those organic molecules that we need? Do we just steal them from other organic molecules from other organisms by eating those organisms? Or do we get it from carbon dioxide and then we have to build those organic compounds from this inorganic carbon? And we have to go through and make organic carbon from this inorganic carbon. We also have nitrogen, and nitrogen can either be come from inorganic sources like ammonia, nitrate, and nitrate and nitrite. And then you can have a few bacteria that are called nitrogen fixers, which can pull the nitrogen straight from the atmosphere, which is typically inert and not going to be reactive to anything, and make them into these inorganic forms of nitrate that can easily be can be more easily be used in the biochemistry of for these cells. So nitrogen fixers, so nitrogen fixers are like what you like people talk about nitrogen fixers with like soil health a lot because plants need nitrogen and not all plants are able to fix nitrogen, just like not all bacteria can fix nitrogen. And so some plants are able to just are only able to get nitrogen from these ammonia, nitrate, and nitrite compounds that have those as like functional groups and things. And if you add nitrogen fixers to the soil, they can start to convert nitrogen gas from the atmosphere into these more usable forms so that the plants that can't have access to it now have more access. The same kind of goes for bacteria that don't have those fixing capabilities, but that doesn't have like a direct, like people don't talk about that. Like you couldn't just wander into that conversation probably on TikTok, for instance. But you might be able to recognize the nitrogen fixers in soil health. So that's why I used it as an example. Anyway, now that I'm done justifying my example, we can also talk about how you get your sources of oxygen, and that source of oxygen usually comes from the atmosphere. Then you have the micronutrients like phosphorus and sulfur, which come from different minerals. So, in the environment around you, essentially. Hydrogen comes from other organic compounds like sugar, has lots of hydrogens. Other compounds have that. Water has hydrogens, and so when you go through when you split the water and through hydrolysis, you can get hydrogens through that. And then it's kind of like, what is the main source of these essential nutrients for these people? And then how do they convert those nutrients into usable carbon? So if they use carbon from other organisms, they can just directly integrate those carbon molecules into themselves and into their own biochemistry very easily. And these are what are called heterotrophs, where they've relied on someone else to do this fixation for them. Autotrophs, on the other hand, they are using inorganic carbon and they need to convert it into organic carbon. And they have to use a chemical source of energy or a source of energy to do this. And this source of energy can either come from light or it can come from the chemicals. And this is what gives you photoautotrophs or chemoautotrophs. And chemoautotrophs can come from things like hydrogen sulfide or iron, redox reactions and things like that that can tear off these electrons and do the biochemistry that's a little bit, the inorganic chemistry that's hard for the biochemistry to do. Then or we utilize light, which does the same thing. We have the energy from the light. We utilize that and leverage that to do work within the cell. We'll talk about those pathways later when we talk about respiration. So we talked about kind of the availability of these nutrients are going to be honestly pretty impactful to a bacteria's growth and survivability, because if there isn't nutrients to be utilized for their biochemistry, they're not going to be able to sustain their biochemistry. They're not going to stay alive. They're not going to be able to sustain their metabolism, you know? When I say biochemistry, I'm talking about just like all of the small little reactions that are happening in pathways that are like, for instance, with cellular respiration, that is a biochemical pathway. And so I'm just kind of like using that term very broadly to just kind of being like all of it that's needed to sustain homeostasis within the cell, you know? So the things that the availability of nutrients is going to like change how readily that bacteria is going to survive, how quickly it's going to be able to duplicate and things like that. Other things are like temperature, oxygen. Oxygen's a nutrient, essentially. Radiations, inhibitory growth factors, things like that will also impact the growth. And then also what other bacteria are around. Maybe it's really crowded, and so there's just like not enough space. And so space availability is also important. Anyway, so with temperature is something that's really important with growth, and it changes how fast these bacteria can grow. So bacteria kind of have a continuum where they can tolerate a wide range of temperatures and grow within them. But there is an optimal temperature where it grows the best at. And there's a minimum temperature where it grows the least, and a maximum temperature where it grows, there's a maximum temperature where it stops growing, and there's a minimum temperature where it stops growing. An example of this in a figure, it's like this, where you have a growth rate, and as you increase your temperature, that growth rate increases to a point, and then eventually it decreases. So you have psychrophiles, which are bacteria that really like cold and can withstand very cold temperatures. You have mesophiles, and these mesophiles are going to be like the room temperature. These are going to be the things that we interact with regularly. Then you have thermophiles, which are going to be really hot and extreme temperatures. And you have extremophiles, which are even more so. And so you can see how psychrophiles have this low temperature preference where extremophiles have this high temperature preference. Excuse me. One thing that's of note is this is in Celsius. Things that you need to know is our freezers are at negative 20. If you're doing science, that's important. If you're doing science with refrigerators, it's 4. For us, our room temperature is 23 and our body temperature is 37. So again, room temperature is 23 and our body temperature is 37. And so you can see how with this mesophile, the optimal temperature is right at body temperature. And so mesophiles are going to be what constitutes most of the bacteria that are commensal on our body, the things that live on us on our day-to-day lives without causing us harm. And pathogens also occur in this range, but in this term, we're just talking about just in general. The reason this growth kind of dips down is because these bacteria have a ton of proteins in them, and proteins are just these very tightly ordered and structured. They're not tightly ordered, sorry. Proteins are well organized and folded chains of polypeptides. And they are folded in a very particular and specific way to give them a specific shape so that they can have a specific function. And so when you increase a temperature of a solution that has molecules in it, at low temperatures, those molecules move slowly. At high temperatures, those molecules move faster. The same thing is happening with a protein. You have a protein with a specific shape, has a specific function. And at certain temperatures, there's some movement. At all temperatures, there's movement, except for absolute zero. But in the cell, there's going to be some intrinsic movement that's happening in this protein, but it's relatively staying the same shape. As we increase the temperature, this protein's going to move more. And it's going to start moving a little bit crazy. And eventually it's going to be in a different shape than it was before. And it's not going to be able to do its function. And this is a process called denaturization, or it's the unfolding of that ordered structure, or that I don't want to say ordered, because when I say ordered, it makes me think of crystals, and crystals are like not what we're talking about. But proteins are folded, and this denaturization is the unfolding of that structure and the loss of their function. And so as we increase their temperature, eventually proteins are starting to lose their function, lose their shape. And eventually it can't sustain that much damage. That's that stress, that's damage on the cell. And eventually it can't sustain it anymore. And so it dies. An example, a really cool example is extreme thermophiles, is they have proteins and they have mechanisms that they have specific proteins that are designed to withstand heat better, but they also have additional heat protective proteins that help correct so for instance, if it was like this and then heat made it like that, there'll be something that's like, oh, well, you should be like this. And that guiding, that chaperoning of the shape kind of protects it from these high heats. And that's why it can survive such high extreme temperatures. So anyway. Then we have oxygen, and oxygen is going to be a very large determinant on where certain bacteria can grow, because oxygen is a powerful oxidizing reagent, and it can kind of wreak havoc on your molecules because of how reactive it actually is. And so so for example, oxygen can be turned into to radicals. It can be turned into peroxides that can then decompose into radicals. And these radicals can then go and attack DNA and damage your DNA. These radicals can attach and like they're essentially really reactive chemical groups, and those chemical groups are just kind of freely floating around in your cytosol. And so if it bumps into the wrong thing that can be compatible, it might accidentally do something it's not supposed to. And it can cause some kind of problem. My apologies. So these, yeah, so some bacteria are able to actually mediate the reactivity or the damage that oxygen can do. And it uses it as like an electron acceptor as like to handle it, or it has certain proteins that handle it. Others do not. And so if it can't handle that oxygen, it will die in an oxygen environment. And so there are some bacteria that just will die in an oxygen environment because of that. So anyway, how we classify them is either by obligate, facilitative, or aerotolerant, where obligate is it must happen. So obligate aerobes would be it must live with oxygen, or it's obligated to live with oxygen. And so this is going to be able to grow in an abundance of oxygen. Facultative anaerobes are bacteria that they can facilitate anaerobic conditions, but they don't love it. Essentially, they can grow. Sorry, I was trying to like relate it like I did a obligate. So facultative anaerobes are bacteria that can use oxygen, but they don't necessitate oxygen's presence. I have an example of this. So here's a tube with liquid where at the top you have high concentrations of oxygen, and at the bottom you have low concentrations of oxygen. And depending on where the bacteria grows, it determines how much oxygen it can withstand. So this first one is at the very top only. You can see that cloudiness, and that's the bacterial growth. And so it can only grow where there's an abundance of oxygen. So it's obligated. It's an obligate anaerobe. Sorry, it's an obligate aerobe. You then have this tube here, which has bacteria kind of halfway. And so it can survive in oxygen and slightly in less oxygen. And this is called a facilitative anaerobe, is that it can live without oxygen, but sorry, I keep confusing myself with this one, so I'm gonna move on. Because I think I don't know what I'm saying that's tripping me up, but I'm just know when I mention this facilitative anaerobe that there's something that I'm saying that's getting hung up. And so I would Google this one and be more cautious when with this term. With obligate anaerobes, these are bacteria that can't live in oxygen. They can't protect themselves from the reactivity of the oxygen. And so the presence of oxygen kills the bacteria. So this last tube is an example of an obligate anaerobe. So it's obligated to live without oxygen. You can see really strong growth at the very bottom where there's little oxygen, but at the high oxygen area, there's no growth at all. This tube here, there's growth all the way through it. This is what's known as aerotolerant, where it doesn't care that there's water. I'm sorry. It doesn't it doesn't care that there's oxygen, but it can handle the damage that it causes, but it doesn't use it. And so this allows it to go all the way through. This is different than the facilitated anaerobe, because the facilitated anaerobe does use oxygen. And because it does use oxygen, it makes it where it prefers more oxygen-rich environments than the air tolerant does. Anyway, sorry about that for any confusion that that caused. Now, we can talk about the binary fission of how bacterial cells go into daughter cells. The process is first you have to grow your cell and start to bulk up. Then you start to replicate the genome. And sometimes you might have accessory plasmids. Sometimes those would be replicated. Sometimes they might not. There might be multiple copies of the same plasmid within the bacteria. And then the cytoskeleton and the cell membrane starts to fuse and create a boundary across that plane of division. And it creates two independent daughter cells. And these two independent daughter cells can either be separated and be independent individual cells, or they can remain connected and get that strepto shape arrangement, sorry. Now, because this is a doubling reaction where one cell turns into two and two turns into four and 4, 8, 16, 32, 60, 420. So these bacteria double usually within minutes to hours. There are some bacteria that do it within days, but that's a lot less common. And usually it's more of like when you have different temperatures. So the lower temperature, usually the longer the doubling time. And when you start a culture, there's plenty of nutrients within that culture. And the bacteria, there's very few bacteria, and they start to grow in what's known as the lag phase. This lag phase is a slow doubling. And they start to kind of get used to the temperature. They start get used to the media and the nutrients and what's available. They start getting all of their transcriptions and things, all of their gene regulations and things like that, all of that, all in order. And then eventually their doubling rate, their growth becomes an exponential growth, and this turns into the log exponential growth phase. Excuse me. And then eventually you're going to start to depreciate the nutrients that are available within this tube, and you're going to start to slow your growth rate. And this is going to slow us down into the stationary phase, where we start to have bacteria that is starting to compete for resources. There's a lot less growth happening. There's a little bit of death that's happening. What's interesting is as your culture gets older and death starts to occur and the waste starts to accumulate, you'll start to see at the very bottom some like dusty, powdery stuff floating bits. And that is debris and cell debris from either dead cells or just waste. And that starts to accumulate within the stationary phase. And then eventually, once it gets too far, major death starts to happen within these cells where they're not just maintaining their population, but they're actually losing population. This is going to start stressing the cells out quite a bit. And so this is where you're going to start seeing like endospores starting to be made and released if they're available, like if they're being made, if they're available to that species. You're going to start seeing a lot more of that debris and things like that. And honestly, your culture, and for most microbiology reasons, you're probably not going to want a culture that's that old. You want to try to take steps to avoid that. However, for certain studies, if you're interested in that phase of resource depletion, for instance, then like perhaps that is what you want. But for regular culturing, you don't really need cultures that are that far gone. And then lastly, we can talk about how pathogens can be used as agents of mass destruction and as agents to incite terror. And so bioterrorism is something that has been going on for an incredibly long period of time, where people would, send sick individuals into cities to kind of weaken those evolved, or in this case, they're talking about throwing bodies through like catapults in the bubonic plague area. But there's a lot more technological and more modern examples as well. That's a lot. They're both scary, but like, I don't know. I feel like throwing bodies just doesn't feel realistic. So like to me, anthrax lace letters feel more of like an impending threat, for instance. Not that that's an impending threat, but it's happened before. But so anyway, uh, something that's happened before is uh, someone had sent out letters to politicians and government officials within the US government and Pentagon and things like that. And these letters had powdered endospores from Bacillus anthracis that they had harvested and put in this envelope so that when you opened it, it would aerosolize this powder. You would inhale it and you would get respiratory based anthrax. Anthrax can cause infection on the skin. It can cause it in the lungs. It can cause it in this digestive tract. And so it is a very potent endospore agent of like infection. And again, it's very resilient. So it's just like overall it's hard to like sanitize for. So um so there are monitoring aspects that have like there are monitoring programs that have come out of these events that occurred to monitor the male for risks of bioterror and biological contaminants. And so these bacteria, if you are evil enough, you could utilize them to create this terror. And there's so many different pathogens that cause different things. There's a lot of different. I'm lost. Yeah, there are. So bad actors are able to utilize these pathogens, not only can utilize these pathogens as like agents of terror, not only as just like, oh, these are health hazards, and they could cause infections in our patients, but these are things that people could weaponize and utilize against people. And so it's important to understand what those risks are to mitigate those possible outcomes. So for instance, understanding that anthrax can be utilized to harvest these endospores and be utilized and weaponized in the ways that it was can help us prevent that from happening in the future. And also can give us ideas of like, oh, that would actually be a really important thing to study on like, how would we like stop that, you know? And so there are many different kinds of pathogens that could be utilized for these as these agents. And so it really comes down to being able to identify that it's happening, being able to, like, by monitoring it, being able to report that it's happening, and then also having a plan to stop it. And so there are lots of surveillance programs um, like uh, rules of order that dictate how we respond to these kinds of threats and um, to how to best prevent them from becoming too widespread. Um, Something that's important is that, for example, so I mean, the last thing I think I'm going to talk about is like anthrax is very specific. So anthrax spores get aerosolized. If you're in that aerosolization sphere, you could be influenced, right? That is targeted. You also have the option that you could also, bad actors could also make the decisions. It's really weird to talk about it like this. Bad actors can also make the decisions to use a contagious agent. What about a hemorrhagic fever like Ebola that nearly kills, has an incredibly high mortality rate, but it has an incredibly high infection rate? Um, or maybe you're wanting to just stifle some something, and so like maybe you're just wanting to kind of hinder the capabilities, and so instead of doing something so lethal, you could do something that's a little bit more innocuous, like maybe you use the flu, but maybe you mix mini strains and use a lot of strains so it's hard to really fight it or something. Like, then people don't die. People might not get sober. They're not going to be like, oh, that's Ebola. That's dangerous. That's a problem. People are looking for Ebola, but are people looking for flu? And so there are people in that have jobs that are specifically working towards answering and solving these potential problems. And it is a true danger with these pathogens and microbes that they can be utilized by bad actors in bad ways, and they need to be understood through research and through policy planning to and public health planning to best handle these situations. I am not an expert in bioterrorism, so I hope that was helpful. Um, with that, I think that that is all of Unit 1. And so I hope that was helpful for you all. I will see you next time. Bye-bye.